Hello, and welcome back to Beyond Networks, the evolution of living systems. We've covered structural and functional perspectives on sort of this minimal process of the evolution by natural selection. And we moved on in last lecture to cover two perspectives that take processes as the fundamental unit of evolution. That was process structuralism and developmental systems theory. Today, we're going to dive into a third process perspective. So we're going to stay on this point here, and that's James Griesemer's reproducer perspective that is based on earlier work by Eörs, Szathmáry, and John Maynard Smith. And so this is an extremely interesting perspective. I've only come across this paper very recently, and it puts so many things, well, in perspective that I want to explain it in a bit more detail. So remember, so the basic sort of motivation here is that the evolution of new traits and new levels of organization, this sort of major transitions in evolution, they require the evolution of new ontogenetic processes, not only the evolution of adaptation. So we need to come up with an explanation of why that is. And I've shown you this quote before, where James Griesemer writes, there can be no escaping the need for an account of how heredity and development intertwine to co-produce conditions of evolvability. We've also seen that perspectives that are based on structural, sort of the units of selection perspective, or functional, the replicator perspective, have problems accommodating evolution. So how can we get past this particular problem? And so let's go back to this sort of major problem of innovation, not just the innovation of new traits, but the innovation of completely new levels of inheritance in evolution. And those are, of course, the major transitions that were first described by John Maynard Smith and Eörs Szathmáry in 1995. Here I'm showing you the revised version by Eörs Szathmáry 2015, where you have 7 major transitions during the course of evolution, from protocells, prokaryotic cells, eukaryotic cells, up to societies with natural language. We're not going to go over those in detail anymore. I just wanted to remind you that sort of that each one of those transitions is characterized by 3 major features. One of them is that it goes from independent replicators at a lower level to higher level units. The second one is it results in some sort of division of labor. We're not going to go into this too much today. And then also it results in the emergence, or is based on the emergence of novel inheritance systems. So we're going to look at point 1 and point 3 in particular, which the reproducer perspective is supposed to address. So the origin of new levels of organization is usually framed in terms of changes in the mode of replication and transmission of genetic information. So units that are replicated independently before the transition now become dependent parts of a higher level replicators. And it's formulated in a very sort of, reductionist replicator inheritor type perspective. You may remember that Maynard Smith is one of the main proponents of that very simple perspective of evolution. So there is a problem with that. If we look at those 7 major transitions, there are not enough different kinds of replicators that we know of to account for so many transitions. In fact, there's only 2 kind of replicators that we know of. So just remember, a replicator is simply defined as something, an entity of which copies are made. Okay, so it's just sort of a copying process. And the only 2 true replicators that fit this definition are genes. That's why Dawkins suggests to focus entirely on a genetic level in evolution, and memes. This is Richard Dawkins' only exception to evolution at the genetic level is the appearance of memes and therefore cultural evolution during the 7th major transition, very much at the end of evolution, when language emerges. Okay, so for all the other transitions, important ones like the emergence of eukaryotic cells or multicellularity, we don't have any replicators that would characterize those specific transitions. So how can we solve this problem? And Szathmáry and Maynard Smith, in a paper in 1997, suggest that these other transitions are connected to the evolution of new reproducers, not replicators. And we're going to spend the rest of the lecture looking into what these reproducers are. Okay, so just to begin this journey, let me sort of stake out the path that we're going to take. And this is basically, the argument is going to try and establish that replicators are just a special kind of reproducer. And I'm going to tell you what a reproducer is in more detail in just a minute. Transition from independent reproducers, transitions happen from independent reproducers to higher level units, not independent replicators to higher level units. And evolutionary transitions involve the emergence of new inheritance systems, which are linked to reproducers, not replicators. So changes in reproduction result from changes in development, unlike changes in replication, which is just a copying process. Reproduction is a developmental process. And I'll fill this account out with a lot of details right now, but these are the main 4 points that you have to remember. So let's start at the beginning and introduce reproducer analysis by introducing sort of John Maynard Smith's and Eörs Szathmáry's idea of what a unit of evolution is. And this will look very familiar. This is from a 1993 paper in the intriguingly entitled journal Abstracta Botanica. And so they define their sort of principles for unit evolution in a way that's very, very similar to what we already know from Richard Lewontin and other people that we've encountered during our journey in this course. So the first principle is there has to be, and this is a little bit different than before, notice that, a process of multiplication. If there is entity A, then it must give rise to more of the same type of entity. There must be heredity. Like begets like. A-type entities must produce A-type entities, and B-type entities produce B-type entities. Crocodiles don't give rise to lions. Variability, heredity is not exact. Occasionally type A objects give rise to type A prime objects, and it may be that A prime equals B, actually. That's just arbitrary, basically. So if different types of entities now have different, have a hereditary difference in fecundity, meaning how many of them, or how fast they reproduce, basically, and or survival, how many of them actually manage to reproduce, then the population undergoes evolution by natural selection. Very similar to Lewontin's 3 principles, but multiplication is worth looking into in a bit more detail. What do they mean by a process of multiplication? It's not just a copying process, because it requires actually a process of development. And James Griesemer defines this idea of development very, very broadly in his paper. So this is important. He defines development as the acquisition of the capacity to reproduce. So a unicellular life cycle, all the metabolic and physiological processes that are going on are development in this sense. So I've called these sort of processes the ontogenetic processes that are required to close the life cycle. I like the word ontogenetic processes because development intuitively is about pattern formation. This is not the case here. So when we talk about development in this lecture, and when you read James Griesemer's paper, development is simply those sort of processes you need to close your life cycle. Any kind of process that contributes to that counts as development here. So development is required to get entities of the same kind at each generation. Remember, this is Robert Brandon's argument that what is actually replicated is not the whole organism. So you need to have some sort of process that restores its organization during each life cycle. And otherwise you don't get the same type of organism. And in this perspective, you treat traits, parts of an organism, as contributing to this capacity, basically, as contributing to development in this sense. Even if they don't immediately contribute to reproduction, their overall effect, from this evolutionary perspective that we're taking now, is to contribute to differential reproduction of the organism. Not development, not in the sense of making form, development in the sense of acquiring the capacity to reproduce. Very different. Okay, so let's look at the particular process of biological multiplication and what that includes. So the first principle here is that biological multiplication is multiplication of material objects. We're talking about multiplication of organisms. Multiplication is not just copying, not just like Dawkins' idea of a replicator. Okay, it not only requires resemblance, which is what defines copying, but also material overlap across generations. You are made of parts of your parents, ultimately. And without that material contribution, you cannot develop. Even Dawkins admits this in some of his writings. You cannot just have informational exchange, you need to have material exchange. So you need some sort of material propagule, which is a material structure that functions in propagating an organism to the next stage of its life cycle, which also can be, of course, reproduction across generations, but not only. So these propagules form the basis of an inheritance system. They ensure that the offspring is born with the autonomous capacity to acquire the capacity to reproduce. I love this. So autonomous, it means you have to be able to develop, acquire the capacity to reproduce without your parents helping you. Well, you know, some parental contributions are acknowledged here, but basically it's your own job to grow up and acquire the capacity to reproduce. And if you don't have that capacity, you cannot evolve, you cannot multiply. It's quite simple, really. But this has a really big and immediate consequence. It follows that there is a dialectic interaction between development and heredity. You don't get one from the other. They entail and generate each other. Now, does that sound familiar? They co-generate each other, okay? They need each other. They are not just recursive, they're also impredicative. One doesn't even exist without the other. So you must have development in your evolutionary account if you're really being serious about understanding reproduction. What do I mean by this concretely? So let's look at this idea of a naked replicator, sort of the selfish gene in Dawkins' most famous metaphor. The idea is that you have, genes are sort of privileged because they make copies of themselves. And this is completely absurd because there is another problem with the concept of replication, okay? And that has to do with variability. So the problem that we encounter here is that this idea of copying is physically impossible. So just copying genes by themselves doesn't work. And I would say that's a rather serious problem that even Richard Dawkins should maybe think about a little more. If your biological account of evolution is physically impossible, that's a bad thing. Remember, I did a big effort at trying to explain the organizational account of the organism, saying it's not mechanistic, but it is completely compatible with the laws of physics and chemistry. Unless we want to become ontological vitalists, this is not a good idea. We want to have an explanation that is compatible with physics and chemistry. And the problem here is, it was first formulated by Manfred Eigen and then elaborated in a beautiful collaboration that Eigen had with Peter Schuster here in Vienna. So Eigen formulated the following paradox, looking at the reproduction of organisms. He said, you know, producing complex enzymes requires a large and complex genome, but replicating a large and complex genome requires enzymes. It's basically Eigen's version of the chicken and egg problem, okay? So how can this arise? It's also impredicative, as you may notice. It's another one of those logical problems in biology with impredicativity. So what you get if you just have a naked replicator is you get an error catastrophe. Basically, the larger your genome that you need to replicate, if you don't have some really, really, really good mechanism to minimize errors, you just degenerate under replication errors. So you cannot be, you're not stable enough to evolve. Remember, you need to be stably inheriting characteristics across generations to be able to evolve. But you can't do that if you're a naked RNA molecule that's just replicating itself, because the errors that you make in copying yourself accumulate over time necessarily. That's the laws of thermodynamics, unless you have some sort of error correcting mechanism. So how do you get this error correcting mechanism? It will be mainly the topic of the next lecture to look at the details of how you get that, but and sort of the requirements you need for that. But right now, it suffices to say that you need some sort of self-organizing process, of course. Okay, and Eigen and Schuster actually suggested a model, a minimalist model for such a process, which they called the hypercycle. And it's basically just a chain of enzymes that not only produce always each enzyme, which is indicated by I1, I2, and so forth in this cycle, not only produces the next enzyme, so catalyzes the production of the next enzyme in the chain, but also catalyzes its own production. So it's a sort of a circular chain of autocatalyzing enzymes. And in principle, if you run this through a mathematical simulation, there's a bit more involved here. You have a sort of a DNA-like molecule making the enzyme that produces, that catalyzes the next element in the model, and so on. But what you get is a sort of a self-reproducing bunch of enzymes, which Schuster and Eigen call the quasi-species of enzymes or catalysts. And this is pioneering work from the 70s, by the way. I mean, one of the earliest models of simple models of a self-making, self-reproducing chemical system. It is not, unfortunately, robust itself. So it also succumbs in the end to an error catastrophe. It helps a little bit, but not too much. So here is the problem, that naked replicators have too much error. They have too little stability. There's too much variability, and we cannot evolve. The other extreme also applies. So Stuart-Kauffman in the 80s then came up with these sort of the idea of networks of catalysts and chemicals that catalyze reactions that maintain themselves. And we've encountered that before when we talked about the organization of the organism. And here are my friends Wim and Stuart, who work on these sort of autocatalytic systems. And they can, in a way, reproduce, okay? Autocatalytic networks can solve Eigen's paradox. They can maintain themselves stably and not succumb to the error catastrophe. But the problem is that they solve Eigen's paradox a little bit too well, okay? Because they don't have to acquire the capacity for autocatalysis. So they don't have to develop. They don't have to acquire the capacity to reproduce. They simply have it. And the problem with that is that it generates too little variability. So heredity will tend, in this case, to be too exact. It is difficult to propagate the variations in autocatalytic systems that are not lethal, that don't break the system, okay? So variation and true inheritance must lie somewhere in between these naked replicators. They must emerge through some sort of developmental process of acquiring the capacity to reproduce. So the job of development in evolution is to create the right range of variability you need for evolution to occur. That's just an additional reason, apart from that reason that you need development to actually produce the organization of the multiplying entity in the first place. You also need it to produce the sort of type of variation that you need to evolve. Remember, this is a problem of the sources of variability, not the consequences. So you need this as a precondition for natural selection to even occur. So it's absolutely fundamental. There is no distinction between proximate and ultimate explanations here. You need this type of proximate process development to get ultimate processes like evolution to work in the first place. And this is, again, a recurring topic in this course. So let me give you a quote from James Griesemer at this point. It is actually possible to make the gene's eye view, Dawkins' view of the replicator work as an analysis of units of replicator evolution, but only insofar as replication, interpreted as a copying process, takes place in host reproducers that serve as interactors. Only that context will provide the right kind of variability and the ability to actually reproduce and sort of re-produce the organization of the organism at every generation. And this is so because the process of replication cannot take place without the development of the capacity to replicate. And nothing in the standard analysis of replicators by John Maynard Smith, by Dawkins, by Hull, and others imply development. Okay, so these sort of perspectives on evolution that ignore development are simply not capable of explaining the emergence of this process of replication. They just take this process of replication, which is very complicated and needs explaining. They take this for granted, which is not okay, because then we don't understand evolution. So let's sort of summarize this argument. So if we come from a very broad perspective, the core of the reproducer perspective is this idea of a multiplier. A multiplier is an entity that produces more of itself, okay? And reproducers are a specific type of multiplier. They're a biological material multiplier. Inheritors, this is a new term, are a type of reproducers, defined as reproducers that form part of an inheritance system. There can be reproducers that don't form part of an inheritance system. So they are not directly involved in propagating characteristics of the organism across generations. Okay, they can work in development or in interaction with the environment. They can have all the necessary features of a reproducer, but not involved in inheritance. Okay, so inheritors are a specific type of reproducer, and then replicators, again, are a specific type of inheritor, which have acquired the ability to copy themselves based on some type of coding process. Okay, that's very important. If you don't have a coding process, you don't have replicators. And so this sort of idea that we encounter with life itself, Schrödinger's account of life, that the basis of life is coding, is just a very specific part of what life is about. It's embedded in this much richer environment of conditions that need to be met for it to work. So basically, the coding process generates a mapping from code source to recipient. The classic coding process in biology, of course, is the genetic code that maps DNA sequences to sequences of amino acids and protein. They're not the foundation of life. They're just a part of this reproducing process. Because there is no such coding process without the process of development supporting it all around. There is no sense in which DNA carries a code without polymerases to read it, or nucleases to replicate it, and ribosomes to translate it into proteins, et cetera, et cetera, et cetera. So there is a code table that represents the genetic code, but behind this code table is a process, a very complicated process of transcription, translation, and also replication of the DNA that is supported by the whole cellular environment. Okay, and that is an essential part of what Griesemer calls development, of course, with his broad definition of development. So development, modern replicators, is deeply embedded in an evolutionary hierarchy of levels of reproductive organization. Without a cell, nothing replicates. Viruses need to hijack cells to replicate. They are just naked RNA and DNA, well, wrapped in some protein, but they cannot replicate without cells, and they're not alive outside the cell. So in the end, replication is a highly context-dependent and highly specialized developmental process. And what's primary here is this developmental process that is part of this dialectic interplay between development and inheritance that gives replication and gives you a life cycle, which is a reproduction, not replication, reproduction, and is part of this life cycle that is the fundamental basis of this process of reproduction and is a reproducer. Okay, so, and this is, if there would be an Oscar for philosophical paper, this sentence would win it completely. My favorite quote this year so far. Far from being master molecules, Griesemer writes, genes are prisoners of development, locked in the deepest recesses of a hierarchy of prisons. Not just imprisoned, but locked into the deepest dungeons of biological organization. And if we claim that this is the fundamental process these are the fundamental units of evolution, we have understood nothing. We have basically put everything upside down. Not that the replicator account doesn't apply in very specific cases, but just very limited specific cases that we have clarified now by using this much broader reproducer perspective. So let's get back to the original motivation. Remember that the reproducer perspective was supposed to be a sort of a generalization of Szathmáry's and Maynard Smith's accounts of major transitions. So what's happening here from the perspective of the reproducer? So evolutionary transitions occur in multiple stages in this perspective. The first is the emergence of a new level of reproducers from lower level processes. For example, if we take the evolution of multicellularity, it would be the emergence of the multicellular life cycle from unicellular, independently reproducing protists. The cells are now reproducing as part of a multicellular life cycle and its own higher level propagules, such as germ cells. So stabilization and maintenance of the new level of reproduction, that's the second step, is required through novel, but that requires novel developmental mechanisms. Okay, so the first step can just emerge. You know, you can get aggregation of cells and then they start to sort of reproduce each other, but that's only stable if you have some sort of mechanisms that block conflicts from below and enforce cooperation from above. So for example, germline soma differentiation is one of these sort of mechanisms in which the somatic cells are policed not to just, you know, reproduce indefinitely on their own. And these sort of policing mechanisms, of course, are exactly what breaks down when you get cancer, which is something we've discussed before. So let's switch. So this is it for multicellularity, basically. This is what happened. Okay, but let's now look at the sort of emergence of natural language and cultural evolution. So what happens here is, in step one, you have the emergence of language, symbolic, abstract concepts, and communication between individuals, which enable non-kin cooperation, and that enables, of course, cultural evolution as a whole, and which is much faster and more flexible than genetic evolution and has had a huge influence in human history the last 10,000 years. And that involves a third step, just like the very beginning of life itself, okay, where genes arise as replicators. So the third step is the evolution of a coding process from new developmental mechanisms. And just like the genetic code arose at the origin of life, more or less, this origin of cultural evolution involves also coding systems such as literacy and numeracy. And abstraction in itself is part of that, or you don't even need to be able to write to have a coding process, so that you have abstract sort of terms for things out there that you can communicate to your fellow human beings, okay? So the reproducer perspective, and this is beautiful, it complements the structural and functional perspectives we were talking about before. It's not competing with them at all. Griesemer writes that it explains how units of selections evolve and at different levels of organization. So it covers the blind spot of the unit of selection structural perspective. It explains the emergence of replicators and interactors through these evolutionary major transitions. And it integrates development as an essential component of evolution, while the structural and the functional perspectives, remember, had problems accommodating development and sort of decided, we're going to talk about that in the next module, to just exclude development from considerations of evolution. So the main point here is that only the structure, function, and process perspectives together provide an exhaustive taxonomy of evolutionary phenomena. These are the words of James Griesemer, of course. And so just like in Aristotle's four causes, remember, you need all four. The four causes are really different types of explanation. And Aristotle believed you need all four explanations, answers to all four questions. The same here, you need answers to the structural question, the functional question, and the process question, and they together provide a sort of understanding of evolution. And I'm going to close with a beautiful quote again. The paper is full of them. I really recommend you read this. I urge transperspective cooperation in the service of comparative analysis. Very important, okay? Don't compete. Take each other serious and then compare. Robustness analysis of the sort I advocate requires patient attention to those research packages one does not adopt, okay, what the other people are doing. I'm taking Hull and Dawkins serious here. I don't agree on a lot of their sort of interpretations of their framework, but I take their assumptions and their perspective very serious because it helps me understand their limitations, and it understands me. It helps me understand my own perspective and its limitations, for the sake of learning about the prospects and limitations of the one perspective to which one has made a primary commitment. Okay, so the entire next module is going to be about discussions that arise because people bash each other's heads in because they have different perspectives. Instead of trying to compare, it's completely insane. The whole of evolutionary biology and a lot of the social sciences are full of such pseudo problems, false dichotomies, and pseudo controversies that are nothing else but the mistaken view that one disagrees fundamentally about something, although one simply has different perspectives on the same problem. Okay, this will be very important. But before we do that, we're going to go back to this idea of the organism as an agent. How does it come into this reproducer perspective? And of course, it comes in really naturally, because what you need to close the life cycle, of course, is organizational closure. That's what we're going to talk about next time. I hope you'll tune in again. As usual, thanks for listening, and goodbye now.